can Protandim help in the management of addictive behavior? And I'm going to talk about a couple of really bad addictive behaviors. Cocaine, which I hope affects nobody in here, and nicotine, which probably has affected some of you in here. And the amazing thing for science is just in the last few years, we've learned how these things work in the brain and what they have in common. And I, for one, was surprised that the mechanism by which cocaine causes addiction is not fundamentally different from the mechanism that nicotine causes addiction. And there have been other papers just published in the last couple of months that extend this, perhaps, to things like food addiction. And if you think about it, they're really not that different. The longing sensation you have for food is a survival value, because if you forgot about it, you didn't get hungry, you wouldn't eat, and eventually you'd lay down and die. So your brain has evolved to keep reminding you of what you need to do and cause you to get hungry and eat, keep your energy levels up. What this story is about is how some chemical substances, some molecules, have learned how to take control of that remarkable system to create their own sense of longing that needs to be satisfied, and paradoxically, to satisfy it acutely. Now, let me show you how this is now understood to work. This is a brain sliced front to back. And you see right there in the middle, there's a red region labeled N accumbens, nucleus accumbens. That has been designated by scientists who study these things as the pleasure center of the brain. And so it controls a lot of what you do, controls things like hunger, things like desire, and satiation from those feelings. In this particular region of the brain that I'm talking about, the nucleus accumbens, that feeling of satisfaction is due to maintaining a basal level of glutamate. And again, this is a common metabolite. The rest of your body's making it. The brain is putting it to a particular use. And so it's been found just recently that cocaine blunts the activity of this region of the brain to release glutamate by downregulating a gene that enables glutamate to get into the cell. And that gene is called cysteine glutamate transporter, sometimes antiporter. And I'll tell you a little more about how that works. Here's a cell, and the yellow protein you see on the membrane of that cell, labeled XCT, is the cysteine glutamate exchange protein. How does that work? It works exactly like a revolving door on an office building. And what it does is it lets cysteine, one molecule, that's an amino acid, into the cell, but it only lets it in if there's a glutamate inside coming out the revolving door at the same time. That's why it's called an exchanger or an antiporter. And we have a lot of proteins on the cell surface that do exactly that. They let things in, let things out. Some of them actively pump things in and pump things out. Others are passive. This one will let cysteine in only if glutamate comes out. Now, the cysteine is very important, and I had questions this morning about glutathione. Glutathione is an important antioxidant. It's one of the antioxidants dramatically affected by Protandim, because among the 500 genes Protandim regulates are the ones seen here in blue, glutathione synthase, that enables the cell to make its own glutathione. So if that enzyme goes up and more cysteine comes into the cell, you can make more glutathione. That cell is protected from oxidative stress. What we're looking at here is the reverse part of that revolving door. Every time a cysteine comes in, which enables the cell to make glutathione, a good thing, this brain cell pumps out one glutamate. And that glutamate is used to maintain a level of satisfaction in the brain. What we found by reading the literature was that cocaine blocks that revolving door. it downregulates that gene. And so if this building, this cell, let's say had 100 of these revolving doors letting cysteine in, glutamate out, after taking cocaine for a month or two, you might only have 25% of those revolving doors still functioning. Okay, that's a concern. How does that manifest? And before we go there, let me say that the surprising part, nicotine, exactly the same thing. Nicotine affects the number of revolving doors the same way. So it decreases your brain's ability to make glutathione and decreases the brain's ability to secrete this important neurotransmitter. So chronically, either nicotine or cocaine, and I'm talking about nicotine because that's what's been published in more detail now, drops the basal level, the baseline level of glutamate, and what that does is it creates a sense of longing. So you see on the left side, normally you have a fairly high glutamate level, and on the right side, that's a situation that's neither longing nor satisfaction. It's just kind of a normal place to be. And if you take hits of nicotine to this region of the brain, and that's shown at the bottom, along the bottom of this axis, and you get repeated exposure to nicotine or cocaine, what happens is the glutamate level starts to drop. This is the baseline level. And that creates a sense of longing, a hunger for something. You need something. And here's the diabolical part. That's, let's say if we look daily or weekly after you start, say, smoking cigarettes, this would happen chronically over that week's time period. If you look at what happens in the short term, the red line shows you that the same exposure to nicotine or cocaine has a short burst of actually releasing glutamate. Okay, so it's doing two things. Chronically starving those cells for glutamate, creating a longing, and acutely, meaning in the case of nicotine, maybe for only 15 minutes, causing those neurotransmitters to be released at the neuron junctions by a different mechanism than the one I just described. So it's affecting glutamate two different ways. It's blocking it in the basal level, and it's stimulating it for a short time. And you can see how diabolical that turns out to be, because taking repeated hits of these substances creates a desire, and the same substance temporarily relieves that desire. So as you move down this curve week after week, your longing increases and you're dependent upon that spike to bring you back up to a level of satisfaction. And what you see here is, about 10 lines down is this cysteine glutamate transporter. It's one of the genes most affected by protandem. And here we see that this, I think, is in brain cells. We've done it in vascular cells as well at two different concentrations of protandem each. And that 6.49 is at a high concentration of protandem. And what that means is it increased the number of those turnstile proteins 6.5-fold, 649% more in the presence of protandem. And this is it in bar graph form, showing you first what nicotine does. And so we're starting with normal, healthy cells, normalizing their level of this turnstile protein that's causing this problem before and after nicotine. And what you can see is the nicotine really represses the expression of that protein. So the cells don't have these revolving doors. They can't get cysteine in to make glutathione. They can't get glutamate out to satisfy your brain. What does protandem do to the same protein? It upregulates it. In this case, at a low dose of protandem in endothelial cells, vascular cells, 250% in the other direction. And so the question raised by all of this is, can protandem help break addictive behavior? And elevating GSH has been shown, on the one hand, to be efficacious in humans trying to break addictive behavior by increasing, or by methods that cause increased glutathione in this region of the brain. And they can break behavior to both cocaine and nicotine. We know that protandim elevates GSH levels, and it's in part by increasing that turnstile protein. And what we found is that, or what has been found by others, is that that turnstile protein is also important in preventing this longing sensation from developing in the brain. So the turnstiles are absolutely essential. And then finally, anecdotal evidence, much of which I gleaned from you at the last Elite Academy, and a lot since then, suggests that indeed protandim might be a useful adjunct, and that clinical trials to really prove this are probably an appropriate question for us to ask.